What is a congener?
A polymer of two or more monomers.
Why does putting salt on the road help to melt snow?
Like we discussed, when a solute is added to a solution, its freezing point is lowered. When salt is placed on snow, it begins to dissolve into any small amount of water on the ice with which it is in immediate contact. This lowers the freezing point of the surrounding water/ice, causing it to melt into the water. This process continues until the salt is completely dissolved.
What is an adiabatic process?
An adiabatic process is a process in which no heat is exchanged with the surroundings.
What is entropy?
Entropy is a measure of the total number of microstates in a system. There have been two widely used definitions of entropy, which were suggested by Ludwig Boltzmann and J. Willard Gibbs. We'll just look at the one specified by Boltzmann, since it's a little more straightforward to understand. The equation for Boltzmann's definition of entropy is S equals kBln. In this equation, Kb is Boltzmann's constant, and is the number of microstates accessible to a system. To get an idea of how entropy works, consider the example of rolling one or more six-sided die. On the first roll, there are 6 possible outcomes. So the entropy associated with rolling one die is kBln, 6. If we roll 2 dice, there are 62 equals 36 possible outcomes, and the associated entropy is kBln, 36. For 3, it's 63 equals 216 possible outcomes, and the associated entropy is kBln, 216. As you can see, the number of outcomes for statistically independent events grows very rapidly, exponentially, with the size of our system, which is also true for molecules. By taking the logarithm of the number of outcomes, we make the entropy scale linearly with system size. While the number of possible outcomes/configurations grows exponentially with system size, the entropy grows linearly, which means that if we double the system size, we double the entropy. This property makes entropy fall into a category of variables known as extensive variables, which just means that they scale with the size of a system in this simple way.
Can elements be converted into one another?
It is possible for atoms of one element to become atoms of another element. One way this can happen is any fission process that results in the loss of one or more protons from a nucleus. the joining of two nuclei to form a single, heavier nucleus is also possible, and this process is known as fusion. Both fission and fusion can result in the creation of new atoms with different numbers of protons than were present before the reaction. These processes are often difficult to control in a laboratory, however. So it's mostly only in specific cases, such as energy production, that chemists and scientists in related fields devote a lot of time to these nuclear reactions.
Why did Eduard Buchner, Germany, win the Nobel Prize for Chemistry?
For his biochemical researches and his discovery of cell-free fermentation. Buchner ground up dry yeast cells, quartz, and a form of silica known as diatomaceous earth, to release the contents of the yeast cells. He then filtered the solution and added sugar. Buchner observed that fermentation of the sugar still occurred in the absence of the whole yeast cells.
What is systematic error?
Systematic errors are unlike random errors in that they change the observed value from the true value in a consistent direction. One example would be if you read a thermometer from the wrong angle. You might consistently read the level of the liquid inside to be a few degrees higher than it actually reads. This would systematically bias the observed value toward a higher temperature than the true value. Another example would be if a scale were calibrated incorrectly, such that it read 5 grams even when no weight was placed on it. This could result in an observed mass that is systematically higher than the actual mass.
What is a fuel cell?
Device that converts chemical energy to electrical energy.
Why did James Batchelor Sumner, United States, win the Nobel Prize for Chemistry?
For his discovery that enzymes can be crystallized, and John Howard Northrop, United States. And Wendell Meredith Stanley, United States, for their preparation of enzymes and virus proteins in a pure form. Sumner was the first to provide concrete evidence that enzymes and proteins could be crystallized, paving the way for further research in this area. Northrop and his co-workers explored the conditions that led to formation of crystalline proteins, and mastered this art, pioneering the way for modern scientists working to crystallize proteins and viruses. Stanley demonstrated that viruses can be crystallized in the same way as proteins, and then showed that viruses are, in fact, proteins themselves.
What are the different groupings for elements on the periodic table?
There are several ways of classifying the elements on the periodic table. One is by the periods, or the horizontal rows, in each of which the properties of the elements change going from left to right. Another common classification is by groups, or vertical columns on the table. All elements in the same group are expected to have similar properties, which is the periodic property, originally noted in the law of the octaves, after which the table is named. Yet another classification is by blocks, meaning the elements are classified by the type of orbital in which the highest energy electrons reside, see below. The logic behind this type of classification is that the type of orbital in which the highest energy electron resides strongly influences the reactivity of the element. Thus elements in the same block usually have similar properties. There are even more ways still of clustering the elements on the table, but these three are the most commonly used.
You might ask then, what is this trans fat everyone talks about in foods?
First, we should point out that most of the double bonds in unsaturated fats are in the cis conformation, where both carbon substituents are on the same side, in nature. Trans fats are fats that have had hydrogen artificially added, which can result in unsaturated fats with a trans conformation about the double bond. There is evidence that these trans fats can be more harmful to your health than other fats.
How does materials science help to improve the fuel efficiency of your car?
One way is through better tires. There are currently tires in development made of rubber that rolls along with less resistance, which has the potential to improve gas mileage by as much as 10% just through the tires. Another way involves using special lubricants that work well at a wider range of temperatures, allowing for better fuel efficiency even when the engine is cold as well as when it's warm. This also has the potential to improve fuel efficiency by about 6%. Additionally, materials science is also the field responsible for developing lightweight materials to construct all of the components of a vehicle, which further contributes to improving vehicle fuel efficiency.
What is a polydentate ligand?
A polydentate ligand is a ligand that coordinates to a metal center via two or more atoms, forming two or more bonds. This resulting complex is known as a chelate complex.
What happens when something biodegrades in a landfill?
Biodegradation describes the process by which microorganisms consume a material and convert it to compounds that are found in nature. This process can happen either aerobically, with oxygen involved in the process, or anaerobically, without oxygen involved in the process. A related term is compostable. This specifically indicates that a material will biodegrade/break down when it is placed in a compost pile.
What is ink made of?
Inks can be very complicated mixtures, but two key ingredients are the pigment used to color the ink and the solvent used to dissolve, or at least suspend, the pigment particles. While modern inks come in every color imaginable for a host of different pen types, historically inks fell into one of two major categories. The first is carbon-based inks. Residues from burning wood or oil, like soot, were used as coloring in these inks, which were suspended in the sap of the acacia tree, known today as gum arabic. The other type of ink used historically is called iron gall. The iron was usually added in the form of iron sulfate, Fe2 plus SO4. Gall refers to gallotanic acid that was extracted from growths, or galls, on oak trees. Iron gall ink slowly darkens as the iron ions undergo oxidation from Fe2 plus to Fe3 plus. The acidity of the ink solution can cause damage to the paper it is used on. So preservation of historical documents that were written with iron gall ink is challenging.
What is a proton?
A charged subatomic particle found in the nucleus of an atom, with a charge equal and opposite to that of an electron.
What is an ultraviolet?
Electromagnetic radiation with wavelengths of 10 nanometers to 400 millimeters. Just shorter than the visible spectrum, but not as short as X-rays.
What is the philosopher's stone?
The philosopher's stone was an object of legend among alchemists. It was believed that the philosopher's stone possessed the power to make gold from other cheap metals. It has also been said to serve as the elixir of life, which was sought by many alchemists. Of course, such a stone has never been discovered, at least not outside of Hogwarts.
What is a d orbital?
An atomic orbital with an angular momentum quantum number of 2.
What is freezer burn?
Freezer burn occurs when frozen food undergoes dehydration due to improper packaging. The humidity level in a freezer is usually quite low, so if food is not stored in airtight packaging, water in the food can be drawn out into the freezer atmosphere by sublimation. Also, because the food is exposed to air, oxidation can occur. Though at the lower temperatures in a freezer, these reactions are quite slow. Thankfully, although freezer burn looks nasty, it's not a food safety concern, it just causes discoloration.
What is progesterone?
Progesterone is a steroid hormone that is crucial to regulating the female menstrual cycle and pregnancy cycle in human females and in some other species as well. It is produced in the ovaries and the adrenal glands and is stored in fat tissue.
Why did Hans Fischer, Germany, win the Nobel Prize for Chemistry?
For his researches into the constitution of hemin and chlorophyll, and especially for his synthesis of hemin. Fischer was interested in biologically relevant pigments, specifically those in human bodily fluids like blood and bile, and the green color of plants. He was the first to determine the correct structure for heme B and heme S, which are iron porphyrin molecules, a large ring with 4 nitrogen ligands coordinated to an iron center. These red-colored molecules help transport O2, and other important functions. He also determined the structure of chlorophyll A, which has a structure similar to that of a heme. But instead of an iron, a magnesium ion sits in the middle of the large porphyrin ring.
How is oil refined?
There are several processes that can be used to refine oil. The oldest way is through distillation, which involves heating up the crude oil slowly, allowing the different hydrocarbons to boil off one at a time, and then collecting them as vapors. There are also newer chemical refinement methods that can use a chemical reaction to convert one type of hydrocarbon into another.
Can there be more than one solid phase for a given substance?
Yes, solids can adopt different types of microscopic arrangements. If there is a repeating pattern to the atoms in a solid, it is called a crystal. If the ordered structure exists for the entire material, then the phase is known as a single crystal. Think of a diamond. If a sample is a bunch of individual crystals, then we refer to the material as polycrystalline. Lots of solids also just have no pattern to the arrangement of their atoms, and this class is known as amorphous.
How does photosynthesis work?
Photosynthesis is the process carried out by plants to harvest energy from sunlight. The most important molecule in photosynthesis is called chlorophyll. It's what collects sunlight and gives plants their green color. Carbon dioxide, CO2, is taken in through cells called stomata. And the plants also draw water up through the roots and into the leaves. The reaction catalyzes when chlorophyll absorbs light and makes ATP along with another molecular energy source called NADP, nicotine adenine dinucleotide phosphate. In the process, CO2 is used up and water molecules are split, releasing O2 gas, which other organisms, like humans and animals, can then breathe.
What is electron microscopy?
An electron microscope is a microscope that uses a beam of electrons to produce an image of a sample. Rather than using light like in a standard microscope. There are several ways of obtaining an image, but the original was the transmission electron microscope, TEM, which produces an image by passing an electron beam directly through the sample. Electron microscopes offer a significant advantage in resolution over traditional light microscopes. This is due to the fact that the wavelengths associated with electrons are much shorter than those associated with visible light. It's possible to achieve resolutions of up to roughly 10 million times magnification using electron microscopes, as compared to about 2000 times magnification in the best light microscopes.
So how little can we detect?
Thankfully, way less than that. Even in the 1990s it was possible to detect lead concentrations as low as 0.1 parts per billion. So analytical chemists are on top of keeping us safe from lead in our drinking water. As long as someone is checking on it regularly.
What are hydrocarbons, and how many different ones are there?
Hydrocarbons, as you might have figured out from the name, are molecules that contain only hydrogen and carbon atoms. There are literally an infinite number of ways to arrange these two elements together. Especially if you include polymers, see polymer chemistry. Hydrocarbons are important molecules. Different sizes and types of hydrocarbons are known as natural gas, gasoline, waxes, like candles, and plastics.
What causes the tides in the ocean?
The ocean tides are caused by the gravitational forces between the Earth, the Sun, and the Moon, along with the centrifugal force imposed by the Earth's rotation. As the Earth rotates, and as the three bodies move relative to one another, the gradual and recurring shifts in the balance of gravitational forces cause the water in the oceans to tend to move toward one coast or the other.
What is an isochoric process?
An isochoric process is a process that takes place at a constant volume.
What are neurons?
Neurons are cells responsible for transmitting information between different locations in the body. Their functions are diverse and include telling muscles to move, passing information from your sensory organs to your brain, making you experience pleasure, and processing information in your brain.
What is the oxidation state?
Indicates the extent of oxidation for an atom, synonymous with oxidation number.
What is the cerebrum?
The cerebrum is the top part of the brain, and it contains your memories, knowledge, and languages, and it manages your senses as well. The cerebrum is the part of your brain that controls your movements and emotions, and it's where your thoughts take place.
What polymers are found in nature?
There are a ton. Proteins, enzymes, cellulose, starch, and silk are all polymers.
What fraction of atoms are empty space?
The fraction of an atom that is occupied by empty space is very large. In fact, over 99.9% of atoms are empty space. The protons, neutrons, and electrons are incredibly small. And the atom occupies such a relatively large effective volume because of the delocalized electron cloud around the nucleus.
What is my credit card made of?
Also PVC, but for the material in credit cards, no plasticizer is added. To manufacture a credit card, usually a few thin sheets of PVC are glued together.
When was Avogadro's constant discovered?
Amedeo Carlo Avogadro published a paper in 1811 describing his theory that a volume of gas at a given temperature and pressure contains a certain number of atoms or molecules regardless of what gas it is. Avogadro didn't actually determine what that number was, however. It took just over 50 years for someone to make progress on that. Johann Joseph Loschmidt, in 1865, estimated the average size of molecules in air. It's nothing short of amazing that he ended up being off by only a factor of 2. Jean Perrin, a French physicist, accurately determined the constant using a few different techniques. He was awarded the Nobel Prize in Physics in 1926 for the work. But Perrin proposed that the constant be named for Avogadro and the name stuck. For more on the use of the constant, see atoms and molecules.
What is the electromagnetic spectrum?
The electromagnetic spectrum describes the entire range of frequencies, or wavelengths, possible for electromagnetic radiation to have. In principle, the spectrum is practically infinite. Though there are limitations on how high or low of frequencies we can practically achieve and work with. On the high end of the frequency spectrum are usually gamma rays, with frequencies of around 1020 Hz. While on the low end are extremely low frequencies of only a few Hz.
What molecular properties do theoretical chemists try to calculate?
Theoretical chemists try to calculate pretty much every molecular property there is. If there's a property we've discussed somewhere in this book, odds are that a theoretical chemist has worked on ways to calculate its value for atoms or molecules.
How did the first matter originate, and why was it all packed together in a very dense state?
Unfortunately, we cannot answer that one, and neither does the Big Bang Theory. Rather, the Big Bang Theory is only focused on explaining the evolution of the universe. From that initial state to what it is today and to what it will become in the future.
What is the freezing point depression?
Decrease in freezing point for a solution from that of a pure solvent.
What determines whether a cell is a skin cell, blood cell, etc.?
The process that new cells undergo to become a specific type of cell is called cellular differentiation. Interestingly, different types of cells don't contain different genetic information, but rather they just express different parts of their genetic information. Signal molecules tell the cell which parts of its DNA to express, which in turn controls what types of proteins and other molecules are present in the cell, and ultimately these factors determine its function.
What is a liquid?
A state of matter with a defined volume, but no fixed shape.
What is physical chemistry?
Physical chemistry is a branch of chemistry primarily concerned with developing a better understanding of the fundamental principles that govern chemical processes. It is an empirical science, meaning that it is based on experimental observations. Though it is probably the most closely linked experimental branch of chemistry to developing new theories in chemistry. As the name implies, physical chemistry is intrinsically concerned with topics in physics that are also relevant to the study of chemistry.
Why did Derek H. R. Barton, United Kingdom, and Odd Hassel, Norway, win the Nobel Prize for Chemistry?
For their contributions to the development of the concept of conformation and its application in chemistry. Barton and Hassel earned the prize for their work surrounding the conformational analysis of molecules. While we draw a molecule on paper in a specific orientation and conformation, there are, in truth, typically many accessible conformations to a given molecule. This is especially true for floppy molecules. And these are where conformational analysis can be particularly insightful and important. Their work drew attention to the importance of rotations and other conformational changes in chemistry, particularly with regard to organic molecules. They showed that reactivity can actually be significantly influenced by the conformation of a molecule, and that conformational changes may be necessary to promote reactivity or allow for a reaction to take place. Why did Louis F.
What is resonance?
Resonance is a way that chemists represent delocalized electronic structure. Let's take that statement apart to understand what it means. Delocalized means that an electron, or a pair of electrons, is not located entirely around a single atom or bond. Take a look at the two structures of nitrogen dioxide, NO2, on the following page. The negative charge is located on one oxygen atom in one resonance structure, but can be found on the other oxygen in the second resonance structure. Notice that we said electronic structure and haven't said anything about atoms moving here. That's because they don't. Resonance only deals with electrons, and the atoms are located in the same arrangement in every resonance contributor. This is important, and makes sense if you remember that the electrons aren't moving from one resonance structure to another. These structures are needed because the actual molecule is more complicated than our simple drawing system can represent.
What are Fraunhofer lines?
When scientists first began observing the spectrum of light reaching Earth from the Sun, the spectrum contained many dark lines, which indicated that light of certain wavelengths wasn't present in the sunlight. These are now called Fraunhofer lines, named after their discoverer. And they are caused by the elements in the outer atmosphere of the Sun absorbing certain wavelengths of light, which prevents those wavelengths from reaching the Earth. Understanding that Fraunhofer lines were caused by atomic absorptions was one of the earliest examples of atomic spectroscopy.
Peterson, United States, win the Nobel Prize for Chemistry?
For their development and use of molecules with structure-specific interactions of high selectivity. These chemists won the prize for developing/finding molecules that can recognize one another so that they react to form complexes in a highly specific way. They investigated the key features of these molecules that allow for such highly specific molecular recognition. In turn, they produced molecules that can mimic the highly specific recognition features of enzymes. This gave way to the area of research that is today known as host-guest chemistry or supramolecular chemistry.
What is nuclear magnetic resonance, NMR, spectroscopy?
Nuclear magnetic resonance spectroscopy is a type of spectroscopy that involves nuclei in a magnetic field absorbing and re-emitting radiation.
What causes the glow in glow sticks?
Glow sticks contain three main components: a dye, diphenyl oxalate, and hydrogen peroxide. The hydrogen peroxide is the chemical you release when you crack or activate the glow stick. Diphenyl oxalate reacts with the hydrogen peroxide to generate a molecule called dioxydenediene. This particular molecule decomposes to release two molecules of carbon dioxide and energy that excites a dye molecule to a higher energy state. To get back to its stable state, the dye releases a photon of light, making your glow stick glow. The particular structure of the dye molecule controls the wavelength, color of this light, which is how you can get glow sticks in different colors.
How are elements named?
The names of elements often have interesting origins. They have been named after people, places, colors, mythological creatures, or for a variety of other reasons. Some are named after scientists, such as curium, after Marie and Pierre Curie. Lawrencium, Ernest Lawrence, Seaborgium, Glenn Seaborg, Mendelevium. Dmitri Mendeleev, Einsteinium, Albert Einstein, and barium, Niels Bohr. Others are named after places, such as Lutetium. Lutetia means Paris in Greek, Californium. Berkelium, Berkeley, California, Americium, Dubnium, Dubna, Russia, Hassium, Hessen, Germany. Yttrium, Ytterbium, Terbium, and Erbium, these last four being named after Ytterby, Sweden. Tantalum, Tantalus, Niobium, Niobe, Promethium, Prometheus, Uranium, Uranus, Neptunium, Neptune. Plutonium, Pluto, Palladium, Pallas, and Cerium, Ceres, are all named after mythological creatures. Though elements can take on different names in different countries, the commonly accepted names are those agreed upon and assigned by the International Union of Pure and Applied Chemistry, IUPAC.
What is a bond length?
The distance separating two chemically bonded atoms.
Are chemists still looking for new reactions?
Yes. Chemistry has hundreds of years of knowledge to build on, but it is in no way a complete field. New ways of making existing molecules, and making chemical structures that are completely new to our planet, are absolutely goals of modern chemistry. Developing new chemical reactions, and understanding old ones, are topics of eternal interest to chemists.
What is an anode?
The electrode at which oxidation occurs.
What is an atom?
Atoms are among the most basic building blocks, making up all matter. The word atom derives from the Greek word atomus, which means that which cannot be split. The existence of atoms, or a fundamental, indivisible unit of matter, was proposed long before modern chemistry and physics came about. It turns out that atoms are actually made up of even smaller particles. But the atom is the smallest unit of matter that defines an element. The smaller particles that make up an atom are positively charged protons, charge-neutral neutrons, and negatively charged particles called electrons.
What is weight?
The force applied by gravity on an object.
What is the atomic radius of an atom?
The atomic radius of an atom is defined as half of the distance between two atoms of the same element held together in a chemical bond. Not surprisingly, these are very small distances. For hydrogen, the smallest atom, the atomic radius is 0.37 angstroms, or 3.7 x 10 to 11 meters.
What is a molecular dynamics simulation?
Molecular dynamics simulations are a computational model of a collection of molecules that interact with one another under a specified set of conditions, temperature, pressure, etc. While electronic structure theory calculations typically only involve one or a few molecules, molecular dynamics simulations can involve hundreds or thousands of molecules all at once. The purpose of molecular dynamics simulations is typically to investigate how molecules interact and react while surrounded by a collection of other molecules. While the energies of individual molecules can be investigated by electronic structure theory, a molecular dynamics simulation allows theoretical chemists to also study how molecules are influenced by their surroundings. These effects can be especially important in liquids, where solvent molecules can have a significant impact on reactivity.
What is random error?
Random errors are variations that increase the standard deviation of a measurement randomly about its mean value. Since random errors should be distributed in a random fashion, both above and below the true value of the quantity being measured, taking repeated measurements can reduce their effect on the measured experimental value. Random errors could be caused by things like noise in an electrical circuit or random fluctuations in the temperature or humidity of the laboratory environment. Random errors are characterized by their Gaussian distribution of values about the mean measured value.
How many bonds can carbon form?
Carbon has 4 electrons available for bonding with other atoms. When carbon is bonded to 4 other atoms, they are arranged in a tetrahedral geometry. These two simple bonding rules have important consequences, as we'll see in this chapter.
What is a spectrum?
A plot of the absorption intensity of light by a sample as a function of wavelength.
How does pectin gel make jelly?
Pectin is a polysaccharide from the cell walls of plants that helps plant cells to grow and also to stick to their neighbors. Commercially, pectin is extracted mostly from citrus peels, but other plants are also used. Pectin is the gelling agent that helps jams and jellies to set, giving them a consistency that is useful for spreading on toast. It does this using a very similar mechanism as the one it uses to keep plant cells together. The chains of sugar molecules, polysaccharides, can form bond between strands, creating an elastic network known as a gel, or jelly.
How many amino acids are found in humans?
There are 20 amino acids commonly found in humans. They are classified into 4 groups based on the identity of their side chain. The 4 groups are polar, nonpolar, acidic, and basic. These 20 amino acids are what make up all of the proteins and enzymes in your body. The interactions between the individual amino acids determine the overall conformation of the protein. Though the relationship between the sequence of amino acids and the structure adopted by the protein is very difficult to predict.
What is crystallography?
Crystallography is the study of the arrangement of the atoms in a solid material. Today, this term generally refers to methods that rely on the patterns of photons. Commonly, X-rays, neutrons, or electrons that are diffracted after impacting a sample. The patterns of the diffracted radiation or particles can be interpreted to determine the structure inside the crystal. The interpretation of the diffraction patterns to yield a chemical structure is by no means a simple task. But crystallographers have been doing this for a long time and it has become a commonplace technique. Crystallographic methods have been used for decades to study the structures of inorganic solids and organometallic complexes. We point out that, while crystallographic methods are commonly used to study inorganic compounds, they have also frequently been applied for studying other types of molecules as well, including biomolecules. While it can often be difficult to obtain a crystalline sample of a biomolecule, such as a protein, crystallography can be extremely useful in deducing protein structures.
What kind of glass is used in your iPhone, or other smartphone, screen?
The glass in most iPhone screens to date, along with that in many other smartphones, is trademarked with the name Gorilla glass. This is an alkali aluminosilicate glass that has been used in over 1 billion devices. It is lightweight, thin, and resists scratching and cracking. Significantly better than many other types of everyday glass.
How do our brains tell time?
Until very recently, scientists presumed, and it's not clear that there were many experiments to support this, that our brain had a stopwatch of sorts built into its machinery. By stopwatch, scientists meant some biological system that created some signal at regular intervals. If this were true, we would be equally good at estimating short and long periods of time. But that's totally not the case. Humans are pretty awful at guessing how long extended periods of time are. Instead, Dean Buonomano at UCLA proposed in 2007 that our brains tell time in a different way. To steal his analogy, imagine a rock being tossed into a lake, which creates a series of ripples in the surface of the water. If you were to throw a second rock in, the ripples from the two rocks would interact with each other. The pattern of this interaction depends on the time between the two rocks hitting the lake. The firing of neurons, the rocks hitting the lake, create these unique patterns in there. Signals and neurons can use the different patterns of signals interacting to tell time between events. The wonderful piece of this theory is that it explains why we're good at telling time over short durations, but not long ones over a long time. The ripples in the water just fade away.
What is fusion?
The joining of two, or more, atoms.
How can I grow a large crystal?
Chemistry principles encountered in this experiment, crystallization solubility materials you'll need. Hot water, from the tap is fine, about 2.5 tablespoons of alum. This is typically found with the spices in the grocery store. It is an ingredient used to make pickles crisp. Its chemical formula is Kal, so for 212H2O, nylon fishing line or thread, about 15 centimeters. A pencil or ruler, 2 jars, spoon, coffee filter or paper towels. The procedure: pour 1/2 cup of hot water into a jar. Add a little bit of alum to the water and stir it in. Try to do this before the water cools down too much. As you'll recall, we can expect the alum to be more soluble at higher temperatures. We want to create a saturated solution of alum, but we also want to make sure that all of it dissolves. Continue adding alum until it will not dissolve anymore. And then you can add a little bit more hot water to get the last bit of alum to dissolve. Cover the jar with a coffee filter or paper towel and let it sit overnight. The next day, pour the liquid into a second jar. There should be small crystals left in the bottom of the first jar. The process of pouring the liquid off of the top of the solid crystals is known as decanting. The crystals left behind will serve as the seed crystals to grow a larger crystal of alum. Pick out the largest crystal, or one with a nice shape that you like. Tie the fishing line or thread around the crystal. And tie the other end of the line to a pencil, ruler, or other long flat object. Adjust the length of the string such that you can use the pencil or ruler to dangle your crystal from the top of the second jar. The one that now contains the liquid, into the liquid without it touching the bottom. Once your crystal is hanging into the solution, all you have to do is sit back and wait for it to grow. This might take several days. If you notice small crystals growing on the sides or bottom of the jar, it would be a good idea to transfer the solution and your single growing crystal into another jar to prevent the alum in solution from crystallizing into several different crystals. This will ensure that you get a nice big crystal. You can feel free to transfer jars as many times as necessary to continue growing your large crystal. History of Chemistry Timeline Year Event 1859 to 1860 The early foundations of spectroscopy are established by Gustav Kirchhoff and Robert Bunsen. 1864 The Law of Octaves. An important step leading to development of the modern periodic table, is first proposed. 1865 The number of molecules in 1 mole of a substance is first determined. 1869 Mendeleev publishes the first version of the modern periodic table, containing 66 elements, and leaving space for yet to be discovered elements. 1869 DNA is first discovered. 1874 The second law of thermodynamics is proposed by Lord Kelvin. 1874 Electric current is proposed to be caused by the motion of electrons. 1876 The concept of free energy is introduced, by Josiah Willard Gibbs to explain chemical equilibria. 1877 Ludwig Boltzmann provides a definition of entropy, along with statistical derivations of several other physical concepts. 1884 Le Chatelier's principle is first introduced, explaining changes in chemical equilibria upon the introduction of changes in a chemical system. 1887 The photoelectric effect is first discovered. 1888 Radio waves are discovered by Heinrich Hertz. 1893 Alfred Werner establishes that some cobalt complexes involve a central cobalt atom bonded to an octahedral arrangement of 6 ligands, laying the groundwork for the field of coordination chemistry. 1894 Noble gases are first discovered. 1895 X-ray radiation is discovered. 1897 The electron is discovered. 1900 Planck's constant is first introduced by Max Planck. 1900 Ernest Rutherford establishes that radioactivity is caused by the decay of atoms. 1901 The Nobel Prizes are awarded for the first time.
What is an elastic material?
a material that deforms when an external force is applied, but returns to its original shape when the external forces are removed.
What is a voltage?
A voltage, or potential difference, is the difference in electric potential energy between 2 points. The voltage describes the amount of work that would need to be done per unit charge, to move a charged object between the two positions. A voltage may be present due to a static electric field, to electric current flowing through a magnetic field, or due to magnetic fields that change over time.
Do reactions always take place at the metal center of an organometallic complex?
Not always. Reactions can also take place at the ligands of a complex. For example, a nucleophile can add to an alkene ligand while it is coordinated to a metal center.
How can we make chemical reactions go faster?
Two of the most common ways to make a reaction proceed faster are to increase the temperature at which the reaction is carried out, or to use a catalyst to lower the energetic barrier to the reaction. With increased temperature, the amount of thermal energy available to overcome the energetic barrier to the reaction is increased. Of course, you'll also get more products faster if you just increase the concentration, S, of the reactant, S. Light can also serve as the catalyst, breaking a chemical bond to produce a species that is more reactive than the original reactant species.
Why did Rudolf A. Marcus, United States, win the Nobel Prize for Chemistry?
For his contributions to the theory of electron transfer reactions in chemical systems. Marcus received the prize for his work studying electron transfer reactions and developing the theory surrounding this elementary chemical process. The transfer of an electron between two molecules is a ubiquitous process in chemistry, and it is important to a wide range of chemical phenomenon, ranging from the conduction properties of materials to chemical synthesis, to the capture of light by plants for the purpose of harvesting energy. Marcus completed much of the work surrounding the theory of electron transfer in the 1950s and 1960s. And he was able to explain the greatly varying rates of electron transfer observed in different chemical systems. Some aspects of his theories were difficult to confirm experimentally, and were not proven until the 1980s, which contributed to why it took so long for him to receive the Nobel Prize for his work.
What is a renewable energy source?
A renewable energy source is one that is provided by resources that are continuously replenished or which will always be available during the foreseeable future of the planet.
How is refined sugar different from raw sugar?
Refined sugar is raw sugar that has been purified by a series of steps that ends with crystallization of a sugar syrup into white sugar crystals. The debate of whether refined sugar is worse for you than raw sugar is ongoing. But chemically refined sugar is just more pure sugar than raw sugar.
What fraction of the oxygen on Earth is produced by the Amazon rainforest?
It is estimated that about 20% of the diatomic oxygen, O2, on Earth at a given time was produced by the Amazon rainforest.
How is liquid extraction used in the chemistry lab?
Liquid extraction is a technique that exploits the differential solubilities of compounds between two liquid phases to extract the compound we want into a single phase. The liquids being used must be immiscible, meaning that they form two separate layers when placed in the same container. The goal is to have our compound of interest dissolved exclusively in a single liquid phase and also to have it be the only chemical compound dissolved in that phase. If this can be achieved, the liquid layer containing the desired compound is then separated and the solvent is removed, yielding the pure substance.
How does hand sanitizer work?
The active ingredient in hand sanitizer is usually an alcohol like isopropanol. This chemical is also used in antiseptic wipes and pads. Because of its ability to kill bacteria, fungi, and viruses.
What is an anion?
A negatively charged ion.
What is alpha radiation?
Alpha radiation involves the fragmentation of the nucleus into two particles. One consisting of two protons and two neutrons, an alpha particle, or in other words, a helium nucleus. And the other consisting of the remaining protons, neutrons, and electrons initially present in the parent nucleus. Alpha decay decreases the number of protons in the nucleus by 2 and decreases the atomic mass of the nucleus by 4 amu.
How is biodiesel produced?
The production of biodiesel fuel involves a chemical reaction called transesterification of lipids. Those from the vegetable oil or fat, with alcohols to produce alkyl esters. Methanol is most commonly used as the alcohol in these reactions, which leads to methyl esters, though other alcohols have also been used. Glycerol is a byproduct of the transesterification reaction, and this compound is actually formed in fairly substantial quantities, ca 10% by mass. This has given rise to research directed toward finding ways to carry out chemical reactions beginning with, or involving, glycerol, such that the cost efficiency of the overall process of biodiesel production might be improved.
What are stereoisomers?
Stereoisomers have the same number of atoms connected in the same order, but differ in their arrangement in space. There are two major types of stereoisomers, enantiomers and diastereomers.
What is a coulomb?
The standard unit of charge, defined as the amount of charge delivered by 1 amp in 1 second.
What is an electric current?
Electric currents are the flow of electrons through a material. The electricity that comes out of the wall in your home or office is just the flow of electrons through a wire.
Why did Archer John Porter Martin and Richard Lawrence Millington Singh, both United Kingdom, win the Nobel Prize for Chemistry?
For their invention of partition chromatography. Martin and Singh won the prize for their development of the basic principles of chromatography, or the separation of chemical substances based on differences in their chemical properties, often their polarity. In their separation method, a drop of a mixture of compounds is placed on a strip of paper, and a solvent, perhaps water or an alcohol, or a mixture, is drawn up the strip of water, resulting in a separation of the compounds in the mixture. This separation occurs because each component in the mixture interacts differently with the solvent. This method has been expanded upon by a large number of later researchers, and chromatographic methods play a very important role in laboratory chemistry to this day.